Hello and welcome again. Today we look at the topic of protein structure, describing these big macromolecules at 4 levels of organization. The primary level, the secondary level, the tertiary level, and in some exceptional cases, the quaternary level of protein structure. But before we go into that, it's really important that we take some time to understand how the unique genome is connected to the unique proteome. And what does that mean? You recall from your previous topics that we all have a unique genetic makeup, which results from the DNA that we carry in most of our cells in the chromosomes which reside in the nucleus. When this DNA is expressed, it unwinds and it's copied, and the process of transcription and translation follows. These processes allow for the unique genome to be expressed as it assembles unique proteins. So there's a connection between the genome, which is unique, and the proteome, which is unique. Organisms have unique genomes, and these unique genomes give rise to unique proteomes. So let's move in and take a closer look at all of this with the use of a model.
Amino acids are often referred to as the building blocks of protein molecules. In most living organisms, there are just 20 of these unique building blocks known as amino acids. Here we represent each unique amino acid with a different style of block. When these 20 amino acids assemble themselves in unique sequences, you get unique protein molecules. Of course, protein molecules carry out a variety of functions in living organisms, including structural roles and functional roles. Of course, roles like hormones and antibodies and enzymes are functional roles, and structural roles are making up muscles and bones and tissues of the body like beaks and nails and hair and skin. Proteins have a variety of function in life, and they are very, very important molecules to understand. And biologists often need to communicate with each other about the structure of proteins. And when they do this, they describe proteins as having primary structure, secondary structure, tertiary structure, and sometimes a quaternary structure. But let's begin our discussion with these 20 blocks. And each of these represents a single unique amino acid. Now here I have a short polypeptide, or a piece of a bigger molecule that would develop into a protein. And what you see here is referred to as the primary structure of a protein. The primary structure of a protein includes all of the amino acids that are present in it and the sequence in which they are linked. So what you would note here is these two yellow blocks represent the same amino acid, and that can happen. And these two white blocks represent the same amino acid. Then you have this one's unique, and all of these others are unique, one-of-a-kind amino acids. So with these 20, and with no limit on the length of the protein chain, you realize that these 20 amino acids are comparable to the letters of an alphabet. And with no limit on the length of a word, it means that we have unlimited types of proteins that we can build. And of course, it's very necessary to be able to do that when you consider that these are the 20 building blocks that give you bacteria, blue-green algae, coconut trees, fish in the ocean, the heart of a human being, and the hoof of a cow. All of these structures are built from protein molecules. And these protein molecules are all built from these 20 building blocks, or amino acids. But because proteins are so important in living organisms, biologists need to describe its structure and understand its structure in great detail. So, in going about this, they describe protein molecules at different levels. So, a particular protein can be described in terms of its primary structure, where all of the amino acids that it contains are listed in a straight line sequence by name. So that might include names like alanine, glycine, lysine, threonine, tryptophan, all of these just listed in 3-letter abbreviations going across in a straight line. That is the primary structure of protein. It tells you what amino acids are present and the sequence in which they are arranged. But once you know that, it's very much like knowing where all of your pieces are on a board in a chess game, or where all of the students in your classroom sit. And once you have that certain layout, it sets the stage for a certain dynamic. And what that means is that the primary structure of a protein is what determines how it folds and bends and twists. And these folds and bends and twists, when we consider what they are and how they change the structure of the protein, we go into higher levels of protein structure. These higher levels are referred to as the secondary structure, the tertiary structure, and the quaternary structure. So here we can see that our protein with its amino acids has bent and twisted and twirled around, and it's formed some alpha helices, some beta pleated sheets, and then it's also twisted into its complex globular form. In some cases, complex proteins join to other complex proteins, and they're held together by groups like iron in the case of hemoglobin. When we describe those proteins at that level, we are talking about their quaternary structure. When we look at the folding, the complete folding of the protein into its final shape, we would be considering its tertiary structure. And when we look at certain aspects of the folding of this complex protein, like the alpha helix and the beta pleated sheet, then we are considering the secondary structure of protein. But to get a better understanding of this, we need to replace our blocks now with molecular diagrams to represent amino acids, so that we will understand the nature of the bonding that brings about the primary structure, the secondary structure, and the tertiary structure.
So the primary structure is very simply the straight line sequence of amino acids. It tells you what amino acids are present in a protein and the order in which they are arranged. So this here is an example of a primary structure. But as we move beyond the primary structure to look at how this long chain of amino acids begins to fold, it's very important that we go back and look at how one amino acid links to another one to form a peptide bond, and how the structure of that peptide bond gives rise to hydrogen bonding. So here we have two simple amino acids. This one is glycine, where this component here is the variable component, or the R group. And in glycine, R is simply a hydrogen. And in the second simplest amino acid, alanine, R is simply CH3. So these are the components that make each amino acid unique. And notice that the rest of the structure of this amino acid is the same. When one amino acid combines with another one, a water molecule is removed, and this is termed a condensation reaction or a dehydration synthesis. Here this hydrogen combines with this OH, and you get H2O being removed. Dehydration or condensation. And then what's left are these components, C double bonded to O and N single bonded to H. This entire thing here is referred to as the peptide bond or the peptide link. And what it does create in the molecule is a C double bonded to an O. An O is a very electronegative element, which means the electrons in the shared covalent bond are held very close to it. So it tends to have a small negative charge. And this nitrogen is also very electronegative. So what it does is it keeps electrons towards itself, and it creates a small positive charge here. And as this long chain begins to build up, what can happen is that because this negative charge here could be attracted to this positive charge here, the entire polypeptide chain begins to take on the form of a spring or a helix with these charged groups, partially charged groups, attracted to each other as the chain develops. It goes into this alpha helix. This bonding has nothing to do with the R groups. It's all about the peptide bond and these groups that have been created. So it's all got to do with what we refer to as the backbone of the polypeptide chain, not considering any interactions brought about by these R groups, which can be variable and a lot more complicated than hydrogen and CH3. So the alpha helix is one type of conformation. Another type of bonding that's associated with this peptide bond and with the polypeptide chain's backbone is referred to as the beta pleated sheet, with a carbon and oxygen here and a nitrogen and a hydrogen here, and then here a hydrogen bond coming up. And similar hydrogen bonds would exist along the length of what is referred to as the beta pleated sheet. The straight line structure that we have, made up of certain specific amino acids that are laid out as a result of a unique genome that lays out a unique proteome. This undergoes coiling of the polypeptide chain into an alpha helix in certain places and twisting into a beta pleated sheet in some places. Once we look at where you have beta pleated sheets and where you have alpha helices, and we understand how they are brought about as a result of hydrogen bonding between the CO group, which has a small negative charge, and the NH group, which has a small positive charge here on this hydrogen. That hydrogen bonding giving rise to the alpha helix and the beta pleated sheet. What we're doing at that point is describing the secondary structure. But protein structure is even more complicated than the primary and secondary structure. The tertiary structure refers to a whole range of interactions that arise out of the unique R groups that each amino acid contains. And these R groups can have sulfur in them sometimes, they can have positively charged groups in them sometimes, they can have groups with lots of carbons and hydrogens which are not highly charged and they're repelled from water, hydrophobic groups, and a range of interactions exist with respect to R. And this is what leads to the coiling and turning of the polypeptide chain into its unique final conformation. When we look at the tertiary structure, it's important to bear in mind that we are considering all of the interactions that arise from these unique groups that each amino acid carries. Because there are 20 of them, and what makes each one different is the unique group that they carry in this section, which we in a general structure refer to as R. So it's all about the R group when we consider the tertiary structure. That's why I've written R here in capital here in tertiary. It's all about the R group. And the R groups interact in a range of ways which are summarized here for you. They interact by having their own kinds of hydrogen bonding, different from the one that arises from the polypeptide chain backbone in the secondary structure. They have hydrophobic interactions here, where groups that don't have charges associate with each other because they're not attracted to anything else around them, so they tend to clump together. A hydrophobic interaction. Then you have ionic bonds, where certain groups that actually carry full charges, negative and positive, get attracted to each other. And then you have full covalent bonds called sulfur bridges in amino acids that do contain sulfur. When they line up in the right position, based on that primary structure, if they end up being next to each other in the right position on the chain, a sulfur bridge, a strong covalent bond could be formed. So you have sulfur bridges, ionic bonds, hydrophobic interactions, and hydrogen bonding. All of these arise from the R groups of each unique amino acid, which are lined up on that sequence, and it brings the polypeptide chain into its unique conformation or its tertiary structure. While most proteins end their structural description at the tertiary level because they're made up of a long chain of amino acids that coil and twist into a complex structure, one single polypeptide chain, there are some proteins that are made up of a coming together, an aggregation of more than one polypeptide chain. Hemoglobin is an excellent example of a protein that combines 4 distinct polypeptide chains, and when this happens, we say that that type of protein has a quaternary structure. So when I say to you that hemoglobin is made up of 2 alpha globin polypeptides and 2 beta globin polypeptides, and each of them are held together by a heme group or an iron group, then I'm describing to you the quaternary structure of that protein. So hemoglobin has a quaternary structure made up of 2 alpha globin polypeptide chains, 2 beta globin polypeptide chains, and each one is held together by an iron group or a heme group. Of course, now that we've looked at primary, secondary, tertiary, and quaternary structure, you might ask the question, what's the point of all of this? Why is it that we need to describe proteins in this way? One example that could show you why we need to do this is the case of sickle cell anemia. Sickle cell anemia can be traced to a base substitution, a change in our genome of a single nucleotide leads to a change in the sequence that lays out the primary structure. One single base substitution changes the amino acid VAL, valine, into glutamic acid. And once this happens, the entire structure of the hemoglobin molecule changes. When the entire structure of the hemoglobin molecule changes and it's exposed to oxygen, it puts strain on the structure of the red blood cells. And the red blood cells in turn change from their regular shape into the sickle shape. So by understanding protein structure, we are able to explain how sickle cell anemia actually happens. Not simply to say that it's a base substitution, that valine is replaced by glutamic acid, but to then explain in terms of the actual structure, how does this substitution, this change, lead to a change in the makeup of the hemoglobin molecule, its conformation. And in studying phenomena like these, biologists need to communicate with each other to describe these phenomena. And by being able to say that the primary structure of protein changes as a result of this, and certain bonds in the tertiary structure are impacted as a result of certain R groups that you have in valine and you don't have in glutamic acid. It aids communication between specialists in the field as they do research and write papers about issues like sickle cell anemia.